Right now, all around us, and yeah, even inside of us, there's this silent, microscopic war going on. See, bacteria are in a constant fight for their lives, and to win, they've developed a kind of genetic superpower. It allows them to evolve at a speed that is just breathtaking. In this explainer, we're going to pull back the curtain on this superpower and see exactly how these tiny organisms are constantly rewriting their own rulebook to survive.
Seriously, just think about it. What if you could learn, say, a new language or how to code an app, not by spending months studying, but just by absorbing the information left behind by someone who already knew how? Sounds like something out of a sci-fi movie, right? Well, for bacteria, this is just another Tuesday. It's how they outsmart our best medicines and thrive in pretty much every environment you can imagine.
So, here's our game plan. We're going to start with why bacteria even need to do this. Then we'll look at what I like to call their genetic sharing economy. After that, we'll take a really deep dive into one specific method they use, called transformation. We'll even check out the actual molecular machines that make it all happen. And finally, we'll see how all of this affects everything from human disease to cutting-edge genetic engineering.
Okay, first things first, why is sharing genes so unbelievably important for bacteria? Well, it all boils down to a really fundamental problem with the way they make copies of themselves. So meet our main character, a typical bacterium. It's a prokaryote, which means, compared to our own complex cells, it's a masterpiece of simplicity. It's got this single, circular chromosome of DNA just kind of floating around in a region we call the nucleoid. No fancy nucleus, no complex little organs, nothing like that. And this streamlined design is great for one thing, replicating super fast. But it comes with a catch. It's missing the gear for sexual reproduction.
And this slide really nails the difference. Eukaryotes, that's us, we use sexual reproduction. We mix genes from two parents, and every single offspring is a unique genetic combo. It creates this huge pool of variation. Bacteria, though, they mostly use something called binary fission. They just copy their DNA and split in two. The result? Perfect clones, genetically identical to the parent.
And right there, that's the massive problem. If you're an exact copy of your parent, and your parent was vulnerable to, say, a new antibiotic or a sudden heat wave, well, guess what? So are you. A whole population of clones is incredibly fragile. One single effective threat could, in theory, just wipe out the entire colony, because there's no genetic variety to fall back on.
This image here shows the concept of selective pressure perfectly. The world is a tough place, right? It's full of challenges that push evolution forward. We're talking fierce competition for food, changing climates, nasty toxins, and of course, the antibiotics that we use against them. In any population that has some variety, when a pressure like an antibiotic shows up, only the individuals who happen to have a resistance gene will survive and reproduce. That's natural selection 101. But for a population of clones, where does that life-saving variation come from in the first place?
Well, to get around this massive vulnerability, bacteria have come up with some truly incredible workarounds to generate the diversity they so desperately need. They didn't just find a solution, they turned acquiring new genes into a high-speed art form.
Okay, so the first method is mutation. Just random typos in the genetic code that pop up when the DNA is being copied. A mutation might be good, it might be bad, or it might do nothing at all. And while it's the ultimate source of all new genes, waiting around for the right random mutation to happen in the right gene at just the right time, it's like trying to win the lottery. It's a really slow, really unpredictable process.
And that brings us to their much more powerful strategy, horizontal gene transfer, or HGT. You should really think of this as a planet-wide genetic sharing economy. Instead of waiting for a lucky mutation, a bacterium can just pick up fully formed, battle-tested genes from other bacteria hanging out nearby, even if they're a totally different species. It's like downloading a software patch that gives you a new ability instantly, instead of trying to write all the code from scratch. And the impact of this is just mind-blowing. Get this, up to 20% of the entire genome of E. coli, one of the most studied creatures on Earth, is thought to have come from these horizontal transfers. That's a fifth of its identity, just borrowed from its neighbors.
So what kind of stuff are they sharing? Well, here's a pretty terrifying example. This is a pathogenicity island from Salmonella. Now, this isn't just one gene. This is a huge, self-contained block of many genes that are all about causing disease. We call them virulence genes. A completely harmless bacterium can slurp up an entire island like this in one go and bam, become a dangerous pathogen literally overnight.
So bacteria have three main ways to pull off this HGT trick.
There's conjugation, which is kind of like direct file sharing, where two cells build a little bridge between them. Then there's transduction, where a virus messes up and accidentally packs bacterial DNA into its shell and delivers it to the next cell. And then there's transformation, which is what we're about to dive into, and that's the ability to just grab bits of naked DNA floating around in the environment.
All right, let's zoom in on transformation. This is the process where bacteria literally scavenge for genetic upgrades from their dead neighbors. It's wild.
You know, this whole phenomenon was first spotted way back in 1928 by a guy named Frederick Griffith. In this absolutely landmark experiment, he was working with two strains of bacteria, a deadly, smooth one and a totally harmless, rough one. So, he injects mice with the harmless strain, mice are fine. He injects them with the deadly strain, mice die. No surprise there. He then takes the deadly strain, kills it with heat, and injects that, the mice live. But here, and this was the huge breakthrough, he mixed the living harmless bacteria with the dead deadly bacteria, and the mice died. The living cells had somehow picked up a transforming principle from the dead ones and had permanently become killers themselves.
But here's the catch. Not just any bacterium can pull this off at any time. To be able to grab DNA from the environment, a cell has to enter this special, highly controlled state called competence. It's an active decision, where the cell turns on a whole bunch of genes to build the specific molecular machinery it needs to find DNA, grab it, and pull it inside. Some bacteria, like the ones Griffith was studying, Streptococcus pneumoniae or Neisseria gonorrhea, are naturally competent, but only under certain conditions.
So, once the new DNA is inside, it doesn't just float around. For it to become a permanent, heritable part of the bacterium's identity, it has to get woven into the main chromosome. This usually happens through a process called homologous recombination. Basically, the new piece of DNA has to find a stretch of the host's chromosome that has a very similar sequence, and then, using a bunch of special enzymes, it just swaps places. This means the whole process tends to work best between bacteria that are pretty closely related, because their DNA sequences are more likely to line up.
Okay, let's walk through the whole play-by-play. Step 1. A donor bacterium dies and falls apart, releasing all its DNA in little fragments. Step 2. One of these free-floating DNA bits bumps into a competent recipient cell and sticks to some protein receptors on its surface. Step 3. An enzyme at the cell surface, a nuclease, acts like a pair of scissors. It chews up one of the two DNA strands, while the other single strand gets actively pulled into the cell. Step 4. Inside the cell, that single strand is protected, and it finds a matching or homologous region on the recipient's main chromosome. And finally, Step 5, homologous recombination kicks in, the old sequence gets swapped out for the new donor DNA, and the transformation is complete. Just like that, the cell has a new version of a gene.
So that's the big picture, but how does the cell actually do all that? Let's zoom way in and look at the incredible protein-powered engine that makes all of this transformation stuff possible.
Let's break down this amazing piece of machinery. What you're looking at is the DNA uptake apparatus, this whole collection of proteins that spans across the cell's outer layers. It's a molecular engine, and it's designed for one job and one job only: to find, grab, process, and import DNA from the outside world.
Now, this machinery isn't exactly the same in all bacteria, mainly because of their basic construction. Gram-positive bacteria, they have a single cell membrane with a thick wall around it. Pretty straightforward. But gram-negative bacteria have two membranes, an outer one and an inner one, with a little space in between. That extra outer barrier means gram-negatives need a more complicated system to get the DNA all the way into the cell.
All right, let's break this down protein by protein, starting with the gram-positives. First, you've got a structure made of proteins like COMGC, which acts like a little grappling hook working with another protein, COMEA, to snag the DNA. Next, a nuclease chops up one strand. Then, a protein called COMEC forms a channel, a tunnel, right through that single membrane. Finally, a powerful motor protein, COMFA, burns some energy to physically drag that single DNA strand into the cell. Okay, now for the gram-negatives. It's a two-step process. First, a huge protein called pile Q forms a gate through the outer membrane. A pilus structure, made of pile E, then helps shuttle the DNA across that middle space. Once it reaches the inner membrane, it looks pretty similar. COMEA binds the DNA, a nuclease does its thing, and a channel protein, COMA, lets it through. Now, what's really interesting is that for N. gonorrhea, scientists haven't yet found a direct equivalent to that powerful COMFA motor protein we see in the gram-positives.
Whew! Okay, that was a lot of molecular nuts and bolts. But now, let's zoom back out and talk about why understanding all this detail is so incredibly important in the real world.
Remember this thing? This pathogenicity island? That complex protein-powered engine we just spent all that time dissecting? Well, that is exactly how a bacterium can acquire a devastating package of virulence genes like this. The ability to perform transformation is a direct pipeline to becoming a more dangerous pathogen, a way to grab new weapons in the unending war against its host.
And look, this whole process isn't just some weird biological quirk. The acquisition of virulence genes through HGT is a primary driver of bacterial evolution. It's how new diseases emerge, it's how bacteria share antibiotic resistance, how they learn to evade our immune systems, and how they colonize new places, constantly creating brand new challenges for modern medicine.
But there is a flip side to this. We scientists have figured out how to hijack this natural process for our own benefit. In the lab, we can take bacteria that aren't normally competent, like most strains of E. coli, and we can actually force them to take up DNA we design. By using things like a quick heat shock, we can make their membranes leaky just for a second, allowing us to slip in custom-made pieces of DNA called plasmids. This is the absolute foundation of modern genetic engineering, and we use it for everything from basic research to producing life-saving drugs like insulin.
And that really leaves us with one final and pretty critical question. We live in a world where these tiny organisms are constantly sharing and trading genetic cheat codes, adopting way faster than we ever could. Knowing that they have this global, high-speed network for trading survival tools like antibiotic resistance and virulence, what does the future of our fight against infectious disease really look like? It's an evolutionary arms race, plain and simple, and understanding these fundamental processes is our absolute best weapon.